Hello, Bruins, and today I will talk about cell theory and history of the microscope. We'll see what inventions led to the cell theory, especially the microscope, and then go a little history of the microscope and development of the cell theory. Okay, now the human eye has a resolution about 100 micrometers, and again, our understanding about biology really depended on what we could see, especially microscopically. And so if you look at this, how big an animal plant cell is, so here's 1 centimeter, and a plant animal cell is between 10 micrometers to 100 micrometers. And then a bacteria is even smaller, and then smaller than a bacteria is a virus, and even very small, it's a small molecule. Now, development of the cell theory was again determined by the invention and advances in microscopy. And here's Hooke's microscope right here, and then you have Leeuwenhoek's microscope. So you heard about Robert Hooke. He's given credit to coin the term cells because they reminded him of a cell, and first to be recorded to see dead cells. So one of the first cells he looked at was a cork, and a cork is like a bark around a tree, and they're used to stop the wine bottles. So this is what he saw when he looked under the microscope. Now, Anton von Leeuwenhoek, he made a very simple microscope, and he just had a ball at looking at everything. He looked at samples of pond, plants, and he saw these little cells, and he called them animalcules. Now, Schleiden in 1838 was given the credit to say that all plants are made up of cells. And then later on, Schwann, think of a swan. Schwann was credited to say, wait a minute, all animals are also made up of cells. And Virchow in 1855 says, wait a minute, all cells come from other cells or pre-existing cells. There's no such thing as spontaneous generation. So the cell theory, first of all, the A is that all living things are made up of either one or more cells. And when we have, do you know the word that means one cell? And that's you probably heard of unicellular. And things that are made with many cells are called multicellular, like a tree. Okay. Now, the second part of the cell theory is that the cell is the basic unit, structure, function in the organism. So here you have plant cells. Plant cells make a tissue. Tissues make a leaf, and leaf is an organ that makes up an organism, the tree. So again, think of the B for basic unit. The cell is the basic unit of all living organisms. And C, all cells arise from existing cells. So think of C as all cells come from pre-existing cells. So C for come from. So cell theory, A, all living things are made up of cells. Cells are the basic units of structure and function in living organisms, and new cells come from pre-existing cells. Okay, so this is like the ABCs of cell theory, and again supported by experimental evidence. Now we can add the modern component, the D for DNA. All living cells have DNA, the hereditary material, and that all living things use energy. All right, so without energy, life would not exist. And the key organs or organelles responsible for energy is chloroplast, again found in plants that do photosynthesis. And then we have mitochondria that does cellular respiration. Okay, so the modern cell theory is D, cells contain DNA, they're hereditary information. E, energy flow of life occurs within the cells. Now, if you remember, as metabolism, all the chemical reactions in an organism. And F, all cells have the same basic chemical composition or formula that we talked about, like lipids, carbohydrates, proteins, nucleic acid. So we know that cells are specialized and they come in different sizes, shapes, and functions. So here we have epithelial tissue that just makes up the skin, our muscle cells, nervous tissue, all in different shapes and sizes. So cell diversity, again, when you look at cells, see the structure versus function, how the structure allows that function. So here's the red blood cells. Main function is it has hemoglobin and it transports oxygen. Our muscle cells are formed so it can allow movement. Our neurons, you see all this branching dendrites that allow nerve impulses to pass through it. Our fat cells has a lot of vacuoles for those fat cells, for those fat vacuoles. So the shape of cells, again, is related to the job. Now, what allowed us to see cells is the microscope. So up here is the eyepiece. Now this is called a compound microscope. This is the kind that we have in our class, and it has 10x magnification. So it's called compound because there's two lenses, 10 plus the objectives. Now the objectives have 3 lenses. The lowest one is what we call scanning, and scanning is something you probably didn't hear in middle school, but we call the lowest scanning, and it has an objective of 4x. The next one is the low objective, and we have that as 10x, and then 40x for the high objective. Okay, so the total magnification would be 10 times 4, 40x for scanning, 10 times 10, 100 for the low, and 10 times 40 for the high. That would be 400x. Now, and when we look at the microscope, we can see depth and focus, and we could tell, look at the thread, and it seems like the blue thread is over the yellow thread. So that's what we mean by depth and focus. And we prepare specimens by preparing what's called a wet mount, where we put a drop of water and a cover slip on our slide. So the one we use in our classroom is called a light microscope. It has a limit resolution about 200 nanometers, and the cells are usually observed can be alive or fixed and stained. So if you remember, you saw the allodia, and you notice that the green things here are chloroplasts, and most of the plant cell is a vacuole that pushes all these cell organelles to the perimeter, and there's a cell wall. Here's an onion cell, and there you see a nucleus, and this dark spot is what we call nucleolus. So any cell that has a nucleus also has a nucleolus, and nucleolus is something, a place where RNA is made. Transmission electron microscope is an electron microscope that basically the electrons are sprayed at the specimen and get scattered, and then that's picked up, and usually it's a 2D picture. The only problem with electron microscope is that the specimen is dead. We never viewed a cell alive in an electron microscope, and notice how big that is. And then we have the scanning electron microscope, and it basically gives us a surface picture and gives you like a 3D view. So if you look at this picture, it has a 3D view, and we basically see the surface of the cell. So again, here's the timeline for major events in cell biology and imaging. So we got Robert Hooke, 1655. 1683, Leeuwenhoek uses his microscopes and see these little cells that he called animalcules. And then we've got Schleiden and Schwann pairing up and propose a cell theory that all plants are made up of cells, that all animal cells are made up of cells. And then later on in more recent times, we've got our first scanning electron microscope and transmission electron microscope.